It's well known when studying the indigenous microbiota of humans that we are unable to cultivate many of the organisms that are present. In the case of the oral cavity, we're far in advance of those scientists who are studying other parts of the body, because approximately 50% of the bacteria in the oral cavity have been cultivated, whereas in the gastrointestinal tract, only about 5 to 10% have been cultivated. So oral microbiologists deserve a pat on the back for having worked so well in identifying these bacteria and trying to cultivate them. There are about 900 different species or taxa in the oral cavity, and we can grow about 450 of these, so it's pretty good. But we don't know what the other 450 species are, what they're doing, or whether they're important or not. So what we did was we used a very innovative approach, the first one that had been carried out on the oral cavity. We cloned the oral metagenome. And this is a technique which has now been widely used. We used this in 2003. The metagenome is the sum of all the DNA at a body site or at any ecological site, you know, the seawater, whatever. Instead of trying to grow the bacteria present in a sample, and we know that we can't grow most of them, then what we do, the modern approach is to extract the DNA from that sample and then identify the genes that are present, and in that way identify what bugs are there and what they're actually doing. So we did this for the oral cavity to see whether we could find other resistance genes in these uncultivable organisms that you don't usually come across. So we took these oral biofilms, we extracted the DNA, we cloned them into expression libraries, and we screened them for antibiotic resistance. So don't need to worry too much about the technical details, but this is the DNA. So you can imagine this DNA contains DNA from organisms which we can't actually grow. So we get this DNA, we break it up into little bits, we stick it into an expression vector, and then we screen out that expression vector, the clones, onto various antibiotics, and we look for antibiotic resistance. And we got 450 transformants which are resistant to antibiotics, and we found the usual suspects, because these are going to include the resistance genes from the cultivated bacteria. But we discovered a brand new tetracycline resistance gene, tetracycline 37. So it's a newly discovered TET gene, and it inactivates tetracycline, but it's distinct from the enzymic mechanism of tetracycline of TETx. So very, very exciting. This is the gene structure. It's got a flavodoxin sort of domain, a metal binding domain, an ADP binding domain, and a tetracycline inactivating domain. So this is a new tetracycline determinant. So quite a scientific achievement, but utterly depressing, of course, because there's another tetracycline resistance gene which you never knew existed. So this metagenomic approach is very, very important for identifying not only antibiotic resistance genes, but new functionalities amongst the oral microbiota. Now, not only are these resistant strains a problem for the treatment of oral infections, but the great worry is that they can be transferred to other pathogens in the upper respiratory tract and the gastrointestinal tract.